Welcome dear student on my channel. Today we'll see about the Mendel's experiment. In this we'll discuss about what is monohybrid cross and what is dihybrid cross. First we'll discuss about the monohybrid cross. Okay, so what is a monohybrid cross? A cross between the parents differing in only one heritable trait is called as a monohybrid cross. So here only one pair of contrasting character is considered. So here we'll take the example about the pure tall and the pure dwarf plant. One of this consider as a male parent, other consider as a female parent. Okay, so cross between pure tall and pure dwarf plant. So for making this, first we'll keep it ready, writing part we'll keep it ready. So here about the parental generation, this is about the parental generation, then about the first filial generation, and then about the second filial generation. So about the parental generation, so what is the phenotype of the parent? So just now I said we have to cross between the pure tall and pure dwarf plant. So what is the phenotype of that? Phenotype will mention here, that is about the tall. So here we'll mention tall is a cross with what? It is a cross with the dwarf. So tall is cross with the dwarf. Then we have to mention the genotype of this particular parent. So what is the genotype of the parent? That is a capital T, capital T. So which is the dominant one? It is a diploid one. Okay. So about the dwarf, what is the genotype? That is a small t and a small t. Always remember here, capital, that is about what? About the dominant character, and this small is there which is about the recessive character. From that, what we get the gamete? So here this is a diploid one, and what is the gamete here? So gamete is a haploid one, so we'll take the single t from this. Same way from here, we'll take the single t, small t. Okay, so from this we got this gamete, and from this we got this gamete. Now here we have to cross. So between this, what the cross is there? So what we get in the F1 generation? That is a capital T and a small t. Capital T and the small t. So by crossing between these two, we got this capital T and the small t. So what it is? It is a hybrid tall. It is a hybrid tall. Okay, so this is what about the F1 generation. Now selfing of F1, we have to do the selfing, self fertilization. So here hybrid tall and the hybrid tall will cross here. Hybrid tall and in the hybrid tall. So self fertilization. This is about what? About the phenotype. Then we'll mention here the genotype of this particular offspring. So what is the genotype? It is a hybrid tall. This one is a genotype. So here we'll mention capital T, small t, and then here the about this capital T and the small t. So this is the phenotype, and this one is a genotype, phenotype. Then this is about the genotype, means what? About the genetic constituents. From this we want the gametes. So here from this we get the two type of the gametes, one with the capital T, other with the small t. From here also we get the two type of the gamete, capital T and the small t. So from here we got this one gamete and this gamete, from here one this gamete and this gamete. Okay. Now in the second filial generation, that is in the F2 generation, these gametes that is considered about the male and this gametes considered about the female. So for male we'll mention here capital T, this gamete here and this gamete here, small t. Same way this gamete and this gamete will mention here, capital T and the small t. So these are the male gametes are there, these are about the female gametes. Okay. Now this one is called as a checkerboard or the Punnett square method. So 1 and 1 from this and this will take one capital T from here, capital T from here. Now this and this, always you have to write the capital letter first. So capital T here and the small t here. This and this, so just now I said you have to mention capital letter first. So capital T and then the small t is here. Now here this and this both are the small t. So here we will mention small t and the small t. So this we got what the result is there, which is a phenotypically we observe. And genotypically if you observe that this one is a pure tall. So this is what it is, a pure tall. This one is a hybrid tall. This one is a hybrid tall. And this one also hybrid tall. And this one is a, what we can say, it is a pure dwarf. Okay, so from this what we get here, we get the phenotypic ratio. Phenotypic means what? From the outside. Outside what we observe here, this one is also tall, this one is also tall, and this one is also tall. So three are there which are the tall. So here that's why phenotypic ratio is a 3. And then about what? About the dwarf, only 1 is there. So from outside, from the phenotypically if you observe that 3 are tall and 1 is a dwarf. So that's why phenotypic ratio is a 3:1. Now genotypically if you observe that it is a pure tall, and these are what? Hybrid tall are there, and this one is what? The pure dwarf is there. So that's why genotypically we get 1, that is a pure tall, and 2 are there which are the hybrid tall. So that's why 2, and 1 is there which is a pure dwarf. That's why it is a 1. So what we get genotypic ratio? 1:2:1. Phenotypic ratio we get 3:1. Okay, so this way we can write about the monohybrid cross. We can take the other example also. Instead of the height, we can consider the shape of the seed. Suppose shape of the seed will be there, it will be a round and a wrinkle. Suppose the color of the seed it will be there, then it will be a yellow and it will be a green here. So only one pair of contrasting character we have to consider, and from that what we got the result? From that we got the phenotypic ratio 3:1 and genotypic ratio is 1:2:1. Now we'll see about the what is dihybrid cross. So what is dihybrid cross? A cross between the parents differing in 2 heritable traits is called as a dihybrid cross. So here the 2 pair of contrasting characters are considered. So we'll take the example, that is a cross of a round yellow seeded plant with the wrinkled green seeded plant. So 2 characters are there. One that is about the roundness is there, other about the yellowness is there. It means about the shape of the seed and about the color of the seed. So here 2 pair of contrasting characters we'll consider. And then after that what the ratio will get, that we'll see. So here first we'll mention the same way how we are done in case of the monohybrid cross. The parental generation here, then the F1 generation, and then about the F2 generation we'll see in the another table. So parental generation, that is about the first. So phenotype, what phenotype is there? We have considered the shape and the color of the seed. So one parent is there which is a dominant, that will mention here. That is about what? About the round and the yellow seed. So this is the one of the parent. This you have to cross with the wrinkle and green seed. Wrinkle and green seeds. So this is about what? Consider as a male, this one consider as a female, or vice versa. Anyway we can consider. So this is about what? About the phenotype of the parent. Now the genotype. So genotype round and yellow. So round and yellow both are the dominant are there. So that's why both capital letter will mention here. So capital R will mention 2 capital R because it is a diploid one. Okay, and then yellowness capital Y. Capital Y. Okay, so this is about what? About the genotype of this. Now same genotype of this is will be the small letter. So small r that is for the wrinkleness, and the small y and the small y for the greenness. Always remember here these are the capital letter, and of course here will be the small letter. So while writing the capital Y and the small y, remember here don't get any confusion in between. Now the gametes. So this is about the diploid. So we get the haploid gametes. So haploid gametes are what? One that is a capital R and the capital Y. So here also the same way, one small r and a small y. From this we got the gametes. From this we got the gametes, which are the haploid one. Now we have to do what? We have to do the selfing. So which this one what we got? After this we got first one. Now we'll do the cross between this. So about the gametes. So this one male gamete, this one is a female gamete, and from that here we got what? We got the first filial generation, that is about the capital R and the small r, capital Y and the small y. So from here we got this one. This is a hybrid one, which is of the first filial generation, right? So here we'll mention which is a round yellow seeded plant. Round and the yellow seeds. Okay, this one is a what we can say, it is a hybrid one. So phenotypically is there, which is a yellow and a round seeded plant. Now here we have to do the selfing. So selfing. So two parents of which consider one as a male and there is a female. So again here the round yellow seed this is the one of the parent is cross with again same round yellow seeds, round and yellow seeds. Okay, so this is the what? This is about the phenotype of the F1. Now from that here we get the gametes. Okay, so after the first we'll find from this here we'll get the genotype. So what is the genotype of this? Same, capital R, small r, capital Y and then small y. This is genotype of this. Same, the genotype of this is a it will be same because it is a hybrid one, capital R, small r and then the capital Y and the small y. Now here you have to write carefully. This is what? This is about the genotype and which is of the diploid. From that we have to get the haploid gametes. So while writing this you have to be careful. So gametes, we'll get the four type of the gametes. We get capital R, then the capital Y, second one capital R, small y, third one small r and in the capital Y, small r, capital Y, and the fourth one will be the small r and the small y. So we got the four type of the gametes from this. Second one, this one is the third one and this one is a fourth one. So these are the gametes are there about what? About the male. Same way we'll get about the female also. Capital R, capital Y, capital R, small y, small r, capital Y and the small r, small y. Okay, so this way we get the four gametes. This gametes about the female. Now these gametes we have to mention where in the Punnett square. This is about our F2 generation, which is a Punnett square or the checkerboard. Okay, so here we'll mention all male gametes and here we'll mention about the all female gametes. What we got? The four type of the gametes in case of the male also, in case of the female also. Okay, so for the male gametes same, we'll just copy here capital R, then the capital Y, this one of the gamete is there, then the capital R, then the small y is there, here the small r and the capital Y is there, and here the small r and the small y is there. These are the gametes of male. Same type of the gametes will be there in the female also. So the capital R, capital Y, capital R, small y, small r and the capital Y and then the small r and small y. So this once we write this gamete, afterward we have to find out the what is the other, what are the offsprings are there in this. So while writing same, how we have performed this in case of the monohybrid. So here we'll get the 4 letters, capital R from here and from here capital R, capital Y, here capital Y. Same way here capital R from here. Always you have to mention capital letter first and then the capital Y first will write, and then this is a small y. So remember the difference between this. Now this capital and this small. So first capital letter and then the small letter. Capital Y. So both are the capital Y. Now from here capital R and this small r. So capital R, small r, and capital Y, small y. So capital Y, small y. Same way here capital R. First we'll write capital R, then capital Y, small y. Here we'll write first capital letter, capital Y, small y. Always remember you have to write the capital letter first. So capital R, both are the capital R there. So capital R, both small y are there. So no need to worry about it. Both small y. Next capital R, small r. So this capital R, small r, then the small y, capital Y. So here we'll mention first capital Y and then the small y. Capital R, small r. So here capital R, small r, then small y, both are the small y's are there. From here this capital R first we'll write, then small r. So capital R, small r, both capital Y. So both are the capital. Next here capital R first and then the small r, capital R, small r, then capital Y first, then the small y, capital Y and then the small y. Here the small r, both are the small r. So small r, both capital Y are there. So capital Y. Next about the small r. So here small r, and first capital Y and then small y. So capital Y, small y. In this case again the capital R, small r, then capital Y and we'll mention small y. So capital Y and small y. Here capital R, small r. So here capital R, small y, small y. Here small r, both are small r there. So small r, capital Y and small y. Always first capital letter and then the small letter. Here both are the which are the small r, small y. Now this we got about the genotype of the next progenies. Now we'll mention here both are letter which are the capital. So it is what? It is a pure one. So pure round yellow. It is a pure round yellow. So this one is a pure round yellow. Here this is a pure one, both small r there and small y. So it is a pure wrinkle green. Okay, so both corner only will get here. Now next here what we get, one capital letter is there, R, capital Y is there. So round yellow, this one is a hybrid one. Here also the round and the yellow. Here also capital letter is there, round and the yellow. So these all are the round yellow. But these are what? They are the hybrid one. Here also round yellow. Here also round yellow. If one is capital, then round, one letter capital Y, then it is a yellow. Here also one capital R is there, other capital Y is there. So again here it is a round yellow. But it is what? It is a hybrid one. Here we observe that, just see this and this way you go first. So round and the yellow. Round and the yellow. Here also capital letter is there. So round and yellow. Now here what we get here, so just remember here this way, this one line is there, this okay, and now go through this like that. Here also capital R, capital Y is there. So round, yellow. Here also capital R, capital Y is there. So round, yellow. So this way we get the triangle of the yellow round. Phenotypically, if you observe that, there are 9. Now this is a R. R for what? R for the round, and the small y is there. That's why it is a round and the green. Round and green. So here one capital R is there. So round, and both y small. So round and the green. Round and the green. Okay. Now here also one capital R is there. Both small y is there. Round and the green. Okay. So here what we get here, 1 and the 2, and here what we get, the wrinkle and the yellow. This one is a wrinkle and the capital Y is there. So that's why it is what? It is a yellow. Here small r is there. So that's why it is again wrinkle, and the capital Y1 is there. That's why it is a yellow. Here small r is there. That's why wrinkle, and capital Y, that's why yellow. Here just now we have observed that. So this triangle is there. We get all yellow round seeds, which are total 9, and this is one is there, 1 which is a round green, this is a round green, and this one is a round green. So 1, 2 and 3 we get the round green, and these are 3. I am making the circle, wrinkle yellow, this also wrinkle yellow, this also wrinkle yellow. So 3 we get the wrinkle yellow. So phenotypically, when we observe that phenotypic ratio, what we get here, the round. So 1, 2, 3, 4, 5, 6, 7, 8 and 9. We get the 9 plants are there, which shows the round and the yellow seed. Next is there about the this round and the green seed. 1, 2 and 3. 1, 2 and 3 we get the round and the green seed. So this is about the 3. These 3 are there, that is a wrinkle and the yellow. 1, 2 and 3. Wrinkle and the green seeds. Sorry, wrinkle and the yellow seeds. 3. And only one is there, which is a pure, which is a wrinkle green seed. So we got the phenotypic ratio 9:3:3:1. Okay. Now we'll see about the genotypic ratio. What genotype is there? So that we'll see now. Now we'll see about the genotype. So first we'll write the genotype of the each one. So this capital R, capital R, capital Y, capital Y. So I'll mention here first capital R, capital R, capital Y, capital Y. Then second one capital R, then capital R, capital Y, small y. Okay. Third one, this is about the capital R, small r, then capital Y, capital Y. Fourth one here, capital R, small r, capital Y, small y. Next is there what we get here. Next, that is about the this one, capital R, small y, small y. Capital R, small y, small y. Next is a capital R, then the this is there, capital R, small y, small y. Capital R, small y, small y. Next is about the small r, capital Y, capital R, capital Y. Small r, capital Y, capital Y. Okay. And next one here we get this is about the small r, capital Y, small y. Small r, then the capital Y, and then the small y. This is about the capital Y. And last one is here, which is a small r, small y, small y. Here we are cancelling just for the practice. We are cancelling with the pencil. So in exam time, no need to cancel. Directly you have to by heart and you have to write the ratio of the same. Now we'll cancel according to this. So here capital R, capital Y, capital Y. So here we got this. So I'll mention 1 here. I'll cancel this. Next, capital R, capital Y, small y. So this is 1 and this is a 2. So this 2. Next, capital R, small r and capital Y, small capital Y. Capital R, small r, this is 1 and this is another 1. So here again 2. The next is there capital R, small r, capital Y, small y. Capital R, small r, capital Y, small y. So this also will cancel. Capital R, small r, capital Y, small y. Here also one more is there, 2, and here also one more is there, this is a 3. Here also one more is there, so this is a 4. So total we got here 4. Next, capital R, small y, small y. Capital R, small y, small y. So this is 1. So we got 1 here. Next, capital R, small y, small y. Capital R, small y, small y. This is a 1. And this one is a 1 more. So here it is a 2. Small r, capital Y. So here is only 1. So 1. Small r, capital Y, small y. So here this is a 1 and this is a 2. So we got 2. And this is only 1 last. So that's why here it's 1. So that's why here we got the ratio, that is 1:2:2:2:4:1:2:1:2:1. So this is about what? About the genotypic ratio. So this everything we have discussed regarding the Mendel's experiment about the monohybrid cross and about the dihybrid cross, along with the example and the checkerboard method. Okay. In description box of this video, the link is given about the number of biology practical videos as well as number of the dissection, that is about the dissection of the heart, dissection of the kidney, dissection of the testes, dissection of the brain. Number of the videos I have already uploaded. The link is given in the description box. So please go through that and share with your friends. Thank you.